continue. Okay, so we're recording the meeting here. So what do you do here? We get this PDF, right? So I'm going to shift it to the other side so you guys can see it. All right, so I made a few pages here. I want to shrink it. Everything is slow now. So I only got those slides here, and basically this one topic, right? So what do? I grab this PDF any day now. Okay. I grab this PDF here. I drop it into the new notebook right there. And now this thing is going to start processing this. And what do? You see these tools over here? Everybody sees those tools? Okay, so one of them says audio overview. So I'm going to do that. Let's see what happens. It will process and it should make a podcast. And this is really cool because the way that that this AI spits out the podcast to you is going to be in the form of a conversation. So you're not going to hear this robotic thing talking in a monotone. You're going to hear basically two people talking about the topic. And this is really cool. I find that the ease of following this type of setting, the attention span is really caught in the way that this AI does that. All right, so this is a tool that you have. Another thing, you can use the other tools like video overview or mind map, but I find that the podcast is one of the most useful ones. The video overview tends to cut out a lot of important information, so I would not rely on that, okay? And this is going to take a sweet time. So this is one of the tools that you can use. I'm not going to be waiting until this thing processes. When you use a different tool like ChatGPT, which, by the way, I do have an example here. Okay. So here's an example of a ChatGPT usage. Okay, let me find the question that I asked. Okay, here. When I was reviewing the subject for model organisms. Okay. So first thing, I remember that there is this statue in Russia that is honoring the work of the lab mouse. So I told the ChatGPT, can you fetch the statue of a mouse knitting DNA? So here we go. The ChatGPT gave me the information of that mouse that is knitting the DNA. Fine. And then I said here, I forgot how long is the mouse generation. Why is it useful as a animal model, as an organismic model? So then ChatGPT spit out what I wanted to know. Okay, so what is the usefulness of the mouse? So I say, okay, generation is about 21 days. And look at this, you have a lot of pups, and usually you can win the pups in a very short time, and they reach maturity like within 6 weeks of life. So ChatGPT is giving me the information that I was not remembering properly, okay, that I needed to refresh. And then I said, okay, fine. Now I need to practice what I know about model organisms. So I told ChatGPT, write some multiple choice questions on model organisms. And there we go. ChatGPT spit out usually about 10 questions. And I use some of these for your iClicker questions. All right. So here I can go through these questions, but you see the way that I have set up here, the answer comes up right away. When you are studying, you don't want to see the answer right away because that's counterintuitive. That's not going to really help you. Right. So you prompt the AI to give you all the answers at the end of your questions. Okay, so you can really practice. The more you practice, the better you get at answering these multiple choice questions. Okay, so this is another tool that you have. All right, so here you see this thing is slow. It takes its sweet time to generate the podcast. So I'm just going to minimize it and finally get to the class so we can cover as much as possible. I'm going to close this. I'm going to just open our cell bio right here, and I'm going to go again to record. And now I don't know where it is. Okay, so here we get back to where we left off. And if the podcast doesn't finish, which probably is not going to finish on time, but I will put that sample podcast in your modules so you can see how that works. All right. So everybody's cool with that? Is that a useful tool for you? Do I see nods? Yeah, yeah. Okay. All right. This is the time that you can actually open your iClicker app and use those reactions that should be enabled. And then you can put some like, thumbs up, thumbs down, question mark, but this we do when I get back. Okay, so that's not, we spend time on that. All right. Now, after you have your, feedback from the AI, whatever you got, make sure that the information you are getting is reliable. You make sure that it does not contradict what you learn in the class and what is written in the PowerPoints. Because a lot of times, unfortunately, AI is not up to speed with whatever we're studying. Okay, so those are tools for you. All right, so here is the slide where we had left off last time, right? We are looking at processes that are common among all cells, right? The smallest living organisms. So we have seen that there are some features that are possessed by all the cells. And here are examples of membrane actions. So here is example of endocytosis, in which the membrane picks up things from the outside and bring it for processing in membrane system inside of the cell, right? So here going inside of the cell is endocytosis, and the opposite process is exocytosis. So export of things from the inside of the cell that goes by exocytosis. So here, when we start asking how is it that cells really became so large and could really take care of a lot of the energetic demands, we know there is this theory that it was only possible because of the acquisition of the mitochondria, right? So mitochondria share a lot of properties with bacteria. Okay, so the theory here is that the mitochondria was actually able to penetrate living a cell, right? And not be digested. It became a symbiose. It went through symbiosis and became a part of the cell. So it's a relationship that mitochondria will provide energy to the cell, and then the cell will provide everything that the mitochondria needs to kind of survive, not on its own, but inside of the cell. And as we're going to see, there are evidences that this was actually what happened, because when you look at the mitochondria, you'll see that they have their own set of DNA. So the DNA of the mitochondria is actually circular DNA, which is, of course, what we see in bacteria. Okay, so they not only they have their own DNA, but some of the genetic code it's a bit different than the eukaryotic genetic code. So, those are very convincing evidences that mitochondria really went through this symbiotic process. And then we see here. So first thing here, so mitochondria, they have their own genome. And the way that they divide is by fission. And then not only they divide by fission, just like bacteria do, but they can also fuse. So you have fission and fusion. And those types of processes really depend on the metabolic demand of the cell, and also how healthy the cell is. Because we know recent studies are showing that a lot of diseases are caused by mitochondrial dysfunction. Especially neurodegenerative diseases are caused by mitochondrial dysfunction. Okay. Another evidence that symbiosis was what happened is that the outside of the mitochondria has this double membrane. Okay, so when you see here, when this bacterium got engulfed and endocytosed, it carried a piece of the cellular membrane surrounding itself. Okay, so mitochondria has the double membrane, and this goes by the similar type of process in which we have seen in the eukaryote, in the other form of bacteria that had engulfed pieces of the membrane to form the double nuclear membrane. So it's the same type of process of membrane process. So here we finally get into the model organisms. So first thing, what is an organism? It is a whole living thing. So if the thing can survive on its own, it is considered to be an organism. Okay, so it can be either unicellular or multicellular. Now, when you start looking at the unicellular organisms, or if you look at bacteria, right, you will know that your prokaryotic bacteria, you have your own model organisms, and E. coli is really the king of prokaryotic studies, right? Now, when you look at eukaryotic cells, you see that the simplest unicellular model that we have is really the yeast, Saccharomyces, Pomba, or something else. So yeast is the model organism, unicellular organism for eukaryotes. Okay. Besides that, you have some other organisms that are easy to study, and we're going to see why. Right? We have model organisms that are going from simplest to more complex. So we go from the worm to a fruit fly to fish, especially like zebrafish. And then you start getting into the world of mammals. So then you're going to have a mouse, and the very, last model organism is the human. But as we are also human, it's kind of difficult to study your own species because as you see, there are some problems with that. Okay. So looking at model organisms of invertebrate, so no spine, right? You see the model organism is a fruit fly. So this is your Drosophila, Melanogaster especially. Okay, so this is a fruit fly. Now, when you start looking at vertebrates, you're going to find two very common model organisms. You're going to find the frog and the zebrafish. Okay, so those do have vertebra. Okay. Then when you look at mammals, you are going to have the mouse as the model organism. Okay. So now what can you learn with model organisms? So here we see in the example of yeast. Okay, so this is just very plain unicellular yeast. And you see that when you look at the genome of this organism, you see that a change in the genome or just a mutation, in this case, a simple gene mutation, changed the form of this organism. So instead of being this bullet form, by changing one nucleotide in the genome, this thing started to look like it's like a T, a mini T-bone or something like that. Okay, so it was found that based on this gene mutation, there was phenotypical change or conformational change to the shape of that model organism. And then when you look at this whole idea of model organisms, we know that the birth of molecular biology and cell biology, they were really founded by studies that were done with this one type of bacteria, which is the E. coli. Okay, so E. coli is shown here. E. coli is this, okay, it's already tiny bacteria, but then if you look at it, the E. coli is also interacting here with this little virus. Okay, so when scientists started to study the interaction of this tiny virus, which is called a bacteriophage, okay, or just phage, okay, with the E. coli, they started to see a lot of molecular and cellular processes that could be studied and manipulated using this model system. So what is it that was discovered using this very simple and tiny model system of bacteria? By the way, where do you find E. coli? Anybody? If you have to look somewhere for E. coli, where are you going to find it? No brave souls in the class today? Yes, yes. I was thinking poop, but yeah, feces. All right. So yeah, it's part of a microbiome. A lot of the E. coli is not harmful. Some E. coli are really, really harmful. You get some that actually can kill you, but you know, a lot of E. coli is just there. Okay, and it's easy to work with it. Okay. So what can you find with the E. coli studies? What was it found? First thing, it was found that DNA was a genetic material of this bacteria. And we're going to see later exactly how the scientists did that, these Hershey Chase experiments. It was really cool, okay? Another thing that was found is how the genetic information was being controlled in terms of expression. So operon models were figured out using this model system. Another thing that's very important was the genetic code. So these bacteria, they really helped figuring out what really was the genetic code, as we're again going to see later on. And besides all that, they're very, very useful for modern molecular cloning because it's very easy to manipulate the small genome of the bacteria. And since the genome of the E. coli is very well known and sequenced, we can really use it for expression of some genes or proteins that can be useful in order to figure out really what the genes are doing or what the proteins are doing, the function of those genes. Okay, so a lot of cellular biology and manipulation of the genome is possible because of this bacteria. And of course, with the sidekick of phages of viruses. All right. So here is the genome representing the genome of the E. coli, and it is circular, okay? And it has one origin of replication, and this goes through theta replication, so it's going to start here and it's going to be bidirectional, and when it gets to the other side, it's going to end over there. So it has one terminus of replication. So simple genome, very much mapped. And those little arrows that you see is really the direction of genes, like how the gene is going to be transcribed. So you see for the genome of the E. coli, you have some genes that are transcribed going clockwise, some other genes are transcribed counterclockwise, but you have a lot of genes that are packed in this small circular DNA. So here is another model organism. So this guy is your yeast, and they are really unicellular, right? And they replicate by, they reproduce by budding. So you see some budding of the yeast. And these guys are very useful because they actually have a nucleus, right? So they have all of the organelles that you see in eukaryotic cells, and they also have a cell wall. Okay, so this is one example of eukaryotic cell that has a cell wall, and it's not a plant. Okay, so it has cell wall, it has Golgi, it has the nucleus here, it has mitochondria. Okay, so there are mitochondria in the cell, and there's a lot of endoplasmic reticulum that is not labeled, but it's right there. So endoplasmic reticulum with the dots, that's the rough ER. And then you see a lot of ribosomes that are floating around. So those are cytoplasmic ribosomes, okay? So you see all of the organelles that are present in eukaryotic cells. You see in this little guy that is your yeast. Okay. So yeast is very useful for making bread. So if you guys like to make bread at home, you buy the active yeast that is live yeast. And when they undergo their metabolism, they produce carbon dioxide, and that's what makes the dough rise. Anybody likes to make bread at home? Yeah, one person. I like to make bread. Yeah, I usually just I never buy bread. I just make bread at home when I'm home. I haven't been home like forever. I'm very much homesick. Okay. Another thing that is useful, that is useful, is to make a beer. You know, like homemade brew beer. Anybody try to make beer? Probably not. I'll give you a little recipe later on how to make your own beer. It might be helpful sometimes. Not before exams, though. No. Assuming everybody's over 21. No. All right, so that's the fun yeast. And now we go to another one that gets a bit more complex. So this guy is the worm. So this is the C. elegans. I don't really think it's very much elegant, but anyway, it's a C. elegans. And the usefulness of this organism, of course, is eukaryotic and is multicellular and is very much mapped. So why is this useful? It's simple. It's only one little squirmy worm. Okay, it's very simple. And it has all of its body has about 1,000 cells. Okay. What is a somatic cell? Is everything in the body, but not a germ cell. Okay, so it has about 1,000 cells, so it's easy to actually count them and to work with them. This worm is transparent. You see through, so you can see the organs and you can map or tag certain proteins and study their function. Okay. We know everything about the genome of this worm. There are about 20,000 genes in the genome of this worm, which is really cool because when you compare it to the number of genes that are encoded in our genome for protein coding genes, we have about 26,000. So a lot of the genes that are present in the worm, they are matching with genes that are in our cells. So a lot of the genes there are homologous to the human genome. So yeah, these worms are actually useful. Okay. Now, the entire lineage of cells of the worm has been mapped. So every single cell is very much, their function is known. Okay. And it's very cool to study nervous system function in this worm because the complete nervous system of this worm has been mapped. It actually has its own cool name. It's called the connectome. And basically, this worm survives with 302 neurons, okay, that have all sorts of synapses among those neurons. So basically, it's a very simple nervous system to study in case you want to see what happens with synapses and what happens in case of certain drugs that you want to alter the function of certain neurons. So this is a very simple organism of the eukaryotic cell that is a lot more complex than your plain yeast that is unicellular. So when you get even to a better level of complexity of complex here, okay, so you're going to have the next one as the frog. So this guy has a vertebra, so it is a vertebrate, right? And is very useful and easy to study the frog. First, because their eggs are so big. You put in the microscope, it's very easy to identify a frog egg. Okay, look at this. It's 1 millimeter. I mean, you can look at a ruler, a regular ruler, and you see the size of 1 millimeter. It's huge, is visible at the naked eye, right? So you can study development, early fetal or embryo development, you can study using the frog eggs. Besides, the development is quite fast. So the embryology here happens quite quickly. So you can study the formation of the organs based on this short period of time. And look at this. This is going by hours. So look at this. Hour 0, you have your fertilized egg. 6 hours later, you already have 16 cells. By 16 hours, you have a blastula. So this is very fast embryonic development. So this is very useful to study the frog for that model. So embryology model and study is done with the frog. And also it's very easy to manipulate the genome and run experiments using this model system. So next one in the scale is the zebrafish. All right, again, it's very good model because it develops very quickly. A lot of the organs of this fish are almost as fast as with the frog. So within 2 days, you have all of the major organs of this fish that is already developed. And again, it's easy to manipulate genetically. And also you have a lot of embryos that can be produced at once. So this is called high fecundity. Okay, so you get a lot of offspring when you're trying to study certain characteristics. Okay. And this is, of course, very important for vertebrate biology and is important for nervous system studies because, again, the entire map of the nervous system is also available for the zebrafish. So here you had neurons and nerves of the zebrafish that had been stained with certain fluorescent dyes. So when you look in the proper fluorescent lighting, you see the entire nervous system of this fish. Okay. So next one, here it is the mouse. So this is the really most important model for mammals. All right, so yeah, mouse, yeah, it's a mammal, okay? And I really like this statue because you see the mouse with trickle or needles, right? And it's really helping to knit the DNA. So the fabric of life here is really, the mouse really has been very, very helpful and allowing us to discover many things, right? So why is it that the mouse is so helpful for mammalians? Well, basically because a lot of the genetic material that we find in the mouse has this homology to the human's genetic material. Look at this, 98% of the genes, of the mouse genes, are shared with humans. So we're genetically very, very similar. And that makes it very important in cases we want to study certain diseases. So we look at the mouse, we see what is the gene that we think is probably causing the disease. And sometimes we can either mutate the gene to see if you mimic the disease, or you can try to use certain drugs to see if you can exacerbate or change the course of the disease by using this model system. Okay, so genetic manipulations that are possible and available to do with the mice. First thing that we can do knockout and knock-in type of experiments. When you do a knockout, that means you look for a gene, you get the gene, you pluck it out of the mouse. So you're going to generate new mice that do not express a certain gene. So that's a knockout. On the other hand, if you have a certain gene in humans that the mouse does not have, you can knock in those genes, and then you study the function in this mouse model. Okay, so you can knock in or knock out. Okay. Besides that, in this example of knock-in, you can make transgenic mice. All right, let's pay attention here. As you know, I never go into politics, but there were some talks that were given by the current administration on mice. There were, I don't think he said transgenic. He said something else. But anyway, make sure that you know what is a transgenic mouse, okay? Transgenic mouse means that you get a gene from a different species, usually human, okay? And you put it in the mouse, so the mouse starts to express a human gene, okay? As a human gene product expressed in the mouse. That is a transgenic mouse, right? Everybody clear with that? All right. Does that sound like a good test question? Is everybody awake? All right, I'm going to ask you. All right. Another thing that you can do with mouse genetic manipulation is this cool technology of CRISPR-Cas9 that will allow you to do very precise gene editing and modification to genes. And yeah, we're going to look at some CRISPR-Cas9 experiments and stuff that is like really, really cool stuff. All right. So very interesting genetic manipulations of your mouse. Another thing that is very useful with this model system is that they have very short life cycles, all right? They like to reproduce a lot. I mean, especially, I mean, think about it. If you have mice in the lab, they are in a cage. They have nothing else to do. If they are lucky, they might have a little wheel that they keep on running around on that wheel and do some and get some exercise. Other than that, what else are they going to do? Not like they're going to be watching YouTube videos or anything, you know. You put a male and a female in a closed cage and with nothing else to do, guess what they're going to do? Do I have to tell you? They're going to produce pups, of course. There's nothing else to do, you know? So they like to do whatever they have at hand, you know, best they can with what they have, right? So they're highly reproducible. They produce a lot of pups. The gestation time is very short, like 20 days, you get a whole litter. And they get a lot of pups at once. In average, you get 10 pups per litter. So you can do a lot of genetic manipulation and have a lot of generations of mice because they are actually by 6 weeks or so they are already able to reproduce. And that's another thing that if you do experiments with mice, you have to always monitor when the litter was born. When you separate the litter, you separate the males from the females. Otherwise, I mean, these guys, they reproduce like black popcorn. And you go back to a cage and you have like, what, 30 mice per cage? Like, what happened here? If you forget about them for a few weeks, then it's like disaster. Okay. So they're very good mammalian organ system. Other advantages of using mice, right? They have very well characterized biological processes. And as I mentioned, a lot of the biological processes are similar to us, to humans. And we can study a lot of diseases based on those biological processes, like cancer or diabetes or cardiovascular or infectious diseases, and even Alzheimer's models or autism models or dementia models. They also have some mice that will behave in similar ways like human. And other very nice types of studies that are correlating are also obesity models. Okay, this is cool stuff. All right. Besides all that, based on decades of studies with mice models, it's very easy to find a type of a model, the type of a mouse model that you'd be interested on. So there are certain companies like Jackson Laboratories that they hold specific mouse models based on what you are interested in. For example, you want to study a mouse that has a tendency to develop Alzheimer's disease. So you're going to ask them for ApoE4 homologous mouse. So the two homologs are expressing ApoE4. So they'll give you can buy the mouse off their shelf. Okay. If you want to study a mouse that has no immune system, they have a mouse for that. Okay. So you ask them for a nude mouse. It's a polite name for the naked mouse, which is a mouse that has no fur. They are kind of ugly looking, and their ears seem a lot bigger than the rest of the mice population. But they don't have immune systems, so you can use that mouse for all sorts of studies. For example, cancer development. The mouse without the immune system develops cancer very easily. So you can implant tumors and study drugs that can kill the tumors using this type of mice. Okay. Besides all that, of course, you have this very extensive genomic database that you can find any gene that is homologous to humans. You can find it in the mouse, and you can base studies in that form with that search. So here in your book, it is showing that a mutation in a certain gene causes in humans this type of discoloration of the skin, and the same mutation on the mouse will cause discoloration of skin and fur. So you can see here that certain mutations are very much homologous. It's a cute mouse. It's a cute baby too. All right, so finally, you're gonna get some points here. So Belle, you ready to press the start button there? So you all know how you do to get your points right with the iClicker. So very easy question for you. Which of the following is considered a first model organism used in cell biology? Okay, so the first
Sorry, I'll have
model,
to disconnect it from the laptop. Hold on.
okay. So first model organism, if you're using the app, I think you don't need the base. So you
Dr.
can,
Hello?
yeah.
I can't get it through my laptop, so I don't think we'll be able to do iClicker today. Can you hear me?
But did you open the iClicker at the site?
Sorry, what?
Did you open the iClicker? Oh, but then you cannot start. Okay. All right. So let's just bank the iClicker for another class. Hopefully by Friday tech issues will be solved in this computer. So okay. So anyway, what is your answer? What is your answer for this question? So you have it ready for next time. Is it E. coli? All right. C. Okay. So here we go with the rest of these chapters, looking at chemical components of the cells and also energy requirements. And I'm not going to read through this because you can read it on your own. And I'm going to really fly through these slides because this is background knowledge and everybody should be very familiar with it. And there's only so much I can ask about that stuff, right? So elements that are found in living organisms, you should be very familiar with those that are highlighted. And of course, you should be very familiar with the periodic table, knowing that to the far right here, you find your noble gases. To the far left here, you find your alkalines. Oh, I forgot the name of this side of the table. Need to refresh. Anyway, find the ones that are highlighted, those you find in living systems. And we know that cells are made up of molecules that are made up of atoms. So basic things about the atom. You have your protons positively charged. You have your neutrons that are not charged. And you have your electrons that are flying around the nucleus, and those are negatively charged. Okay. So the atoms has equal number of protons and neutrons, except for the hydrogen. Hydrogen only has a proton. It does not have a neutron. Hydrogen has proton and electron, no neutrons. Okay. Okay. There we go. Hydrogen is a very unique thing. And then very brief review on electron shells. All right. So you're going to fill up shells that surround the nucleus with certain number of electrons, and every shell can carry a certain number of electrons. And just quick review, right? The electrons that are going to fill out the outmost shell, those are called your valence electrons. So you fill up the early shells first, and then you go to the next higher shell that can be filled up. And I'm not going to ask you if you remember those shells. 1s2, 2s2, 2p6, 4s2. I don't even remember. I don't mind if you remember or not. Just remember that you have shells that are holding electrons. All right. And the outmost shell, those are called your valence shells. Most of those are partially filled for the elements that are important for life. That means they can react with something else. So the outmost shell electron is going to allow reactions because if they are not totally filled, then they are less stable than an atom that has those shells filled. All right. So what happens here? If you have, for example, the simplest atom here, hydrogen, that has only one electron. So the first shell is partially filled. So this thing is it has a high capacity of reaction. Okay. Second, yeah, of course, my husband has to call me when I'm teaching. He doesn't know my schedule yet. All right. So here, if you fill up an entire shell, you are going to get a very stable atom. So every time you fill up a shell, you end up with a noble gas. Look at that. Helium, filled up shell. Neon, filled up shell. Argon, filled up shell. So every time that you fill up and you have one extra electron, you end up with an atom that is highly reactive, like potassium. So this guy has only one electron in the outermost shell, and he doesn't like to have it there because he wants to have all shells filled up and he wants to behave like a noble gas. So this guy here, if it can, this guy is going to kick out that electron to look like a noble gas, and then it becomes less reactive. So very, very brief review of chemistry. Okay. So here, what is it that what are the things that are very important for the reaction of those electrons, right, in the outermost shell? They can share electrons and make covalent bonds, or they can make ionic bonds, okay? So first thing on covalent bonds, what's going to happen? The atoms are going to get very, very close together, and they're going to share electrons to fill up their outermost shell or valence shell. And by sharing the electrons, they are going to make up a molecule. So that is your covalent bond. Okay, so those covalent bonds are very, very stable, and they have certain characteristics. So when you form covalent bonds between two atoms and is a single bond, that bond has the ability of rotation. So you can move the rest of the atom around that single bond. On the other hand, if you form molecules that have double bonds, not just a single bond, that molecule is going to have some other characteristic. Like, for example, it's going to not rotate, so it's going to be a stable, flat type of bond, as we're going to see later when we look at peptide bonds. Okay, so these guys are not flexible, and they are also stronger than the single bonds, because you see here, double bonds tend to be shorter than single bonds. So they don't rotate, and they're stuck in a plane, and they are stronger than single bonds. It is possible for certain atoms to make three bonds, like carbon can have triple bonds, but you're usually not going to find triple bonds in living systems. It's very difficult to find any triple bond in a living system. Okay, here are some examples of triple bonds that molecules that have triple bonds. Okay, so here a brief review of covalent bonds, so you can read that on your own. Okay, so here, this is important. Not all bonds are equal. Okay, so some bonds are going to be stronger than others, and then some bonds can even have more energy than others. Okay, I was going to see later on what kind of energy are we talking about, right? So you have some covalent bonds that are weaker, and if the bond is weaker, it can be broken easily. So when you break bond, you can donate energy that is stored within that bond. Okay, so less stable bonds are easy to break, and they can easily donate energy. All right, so we are going to stop here because already 4:17. And we will pick up on Friday from this, because after this, we're going to get into water properties, and that's a really cool thing to look at. Okay. And by Friday, let's see how much I can cover. And depending on how much we're not going to be able to cover, I will add it to videos for you to catch up with. So I'm going to hang around here for some more time. If you want to come up to the front and chat with me for, for any reason, just like I do in the regular class.
Hello, Professor.
Yes.
Can you hear me just fine?
Yeah.
Okay. Hello, I'm Stephanie Abraham. I'm a student and I had a question about the lecture.
Okay.
When you were describing in the example of like the potassium, can you say why it's highly reactive?
Because it has one more electron on the outmost shell, and the electron is by itself. And if it loses that electron, it will behave like a noble gas, which is very stable. So if it holds on to that electron, it is very unstable because the electron is like far away from the nucleus and is just alone in that outer shell. Okay? And what happens is that you could add some more electrons there, but because of the valence of the outermost shell, it's like because you would need to put a lot more electrons there to give it stability to that outermost shell, and it's more difficult to do that because of the positive charges in the nucleus. There are only so many negative charges you can add to that atom. Okay, so when you look at the periodic table, you will see that in order for the stability of the outer shell to be reached, sometimes it's easier for the atom to lose an electron, or some other times it's easier for an atom to gain an electron. So if the atom is closer to the right side of the periodic table, it's easier, it is getting closer to filling up the outermost shell. So it's easier for the atom just to gain an electron, to steal an electron from somewhere else and to fill up its outer shell. So when the atom is able to do that, you say that the atom is electronegative. So it will attract electrons to itself. Okay? So gaining electrons is going to make the atom more stable. And that's the case of oxygen. Okay? And fluoride. Okay? Now, if you look at the atoms that are closest to the left side of the periodic table, it's easier for them to get rid of their outer electron and to become more stable. So that's the case of the alkaline metals. Okay.
So on the left side of the periodic table, it's easier to get rid of, and on the right side, it's easier to gain an electron.
Yeah. So the electronegativity of the periodic table, it goes to the right and up. Okay, so let me see here. If you go look back here at the periodic table. Oh, I never stopped sharing. All right, so you see here the periodic table.
Yeah.
Okay, so here the electronegativity, it will go that way and up. So the more to the right side, the more electronegative the atom will be. Okay, so when you look here at these atoms that are very important for life, carbon, nitrogen, oxygen, fluoride here, you'll see that nitrogen is more electronegative than carbon. Okay, oxygen is more electronegative than nitrogen. Okay, so these guys here to the left, not the noble gases, but all the way to this side here, they are more electronegative than whatever comes to the left. Now, if you go from down to up, you will see the electronegativity goes up that way. So oxygen is more electronegative than sulfur. Okay?
Mm-hmm.
And then sulfur is more electronegative than selenium. Okay, so it goes up that way. So your electronegativity goes this way and up. Okay? So if your electronegativity goes that way, so these guys at this side over here, they really want to get rid of that extra electron they have. So then they will behave like noble gases. So these guys here do not react at all. Okay, so the noble gases, you see that nothing is highlighted here. They don't react at all. They don't like to react at all. Okay? So
Okay.
for example, helium here is highly unreactive. And yeah, it is a gas, right? It's like to stay on its own. It's not going to make molecules. It's just playing by itself. Okay. And helium is a very safe gas that we use like to fill up balloons, for example, right? When you compare with another gas that is hydrogen, okay, so hydrogen is a gas, but is highly reactive. It has that outer electron. So if you press on hydrogen, it is going to react. If you have a spark of electricity in a ball of hydrogen, it will react by exploding. Okay? So although it is a gas and is also very cool, that is lighter than oxygen, you can fill up balloons with hydrogen, but if you walk across a room and you get electrostatic energy that transfers to that balloon is going to explode. And there was a case, I think it was in the 50s, of this huge, what do you call them? These huge balloons that carry people, like you see them sometimes in Florida, they're like close to football games, like
Yeah.
directable, what do you call them? I don't even know.
The blink, I think.
Yeah, that thing. So there was a case, I think in the 50s, that somebody had the smart idea of filling up one of those with hydrogen instead of helium. And the whole thing exploded. And you can imagine that was like, big disaster. I think that was a Zeppelin or something like that. Yeah. So highly reactive atoms, not reactive atoms. Okay.
Okay, and I had another question. I wrote down something of like weaker bonds are broken easier, and then I put stored in bond easier.
Yeah. So when we
Is
look,
that right?
yeah. So when you look at the weak bonds, okay. So we see that because it's easier to break them,
Mm-hmm.
you can harvest the energy that they hold, as we're going to see in case of ATP that has the three phosphates. So in that case, you have like a phosphate close to another phosphate, close to a third phosphate. And then you see what are the charges that are in each of those phosphate groups. And you see that they're really packed with negative charges. So you have a ball of negative charge connected to another ball of negative charge, connected to a third ball of negative charge. And it's a lot of negative charge together. So the bond between the last two phosphates, that bond is kind of weak because it's holding together stuff that is negatively charged. So it's because it's easier to break that bond and click off one of those phosphate groups, we say that that bond is a high energy bond. It holds energy that can be harvested in an easier form. Now, when you look at the first and the second phosphate, that bond is also high energy, but is not as high as the second and third phosphate. Okay, so not all bonds are the same in a matter of storing energy. All bonds do, covalent bonds do store energy, but if you cannot break them, you cannot harvest the energy. So it's not useful energy.
Okay. And earlier you said that for a specific site that we only needed to remember that we have shells in the electrons.
You have electrons in the shells.
Is it shells or electrons in the shell?
Well, what is holding the electrons are the shells. So
Mhm.
here, okay, so if you look, for example, here, okay. So here is showing carbon atom. So you have shell, it's only showing two shells here, okay? So you have one S2 and one P4. So the name of the shells, they go like S, P, D, something like that, right? So that's
Mhm.
all like basic chemistry. So the first shell in this carbon, the first shell is filled up. So you have two electrons and only two electrons will fit in this shell, okay? So these two electrons fill up the shell. That shell is stable. Now the outmost shell has 4 electrons, but it can hold 6, right?
Mhm. Yeah.
Is it 6? Yeah, from what I remember properly. Yeah. So here, now what is going to happen here? What is going to happen is that 6 or 8, 1, 2, 3, 4, or 8. See, I forgot the basic chemistry too. What happens in this outermost shell, you have these electrons that they're not really happy. They have this carbon will have to get more electrons in order to stabilize the outermost shell. Okay. And the way that is going to find the stability is by getting close to another carbon molecule and share a couple of electrons. When it shares a couple of electrons, that is considered a covalent bond. So it's going to share electrons all the way to fill up that outmost shell, okay? So how many shells do you go? How many electrons? 1, 2, 3, 4, 5, 6, 7, 8. 8 in this outmost shell that is the second shell in here, okay? So 8 of them is going to fill up the shell, and it's going to make carbon to behave like neon, okay? Neon is a noble gas. All right, so let's see what happens here. Carbon only has 4 of the 8 possible electrons, okay?
Mhm.
It's not going to donate these 4 electrons, otherwise it's going to be very electron deficient. It's not going to hold positive charge of 4, okay? So what it is not going to attract or steal electrons from something else, otherwise it's going to have negative charge of 4 and it's supposed to be neutral, okay? So what it's going to do is going to start sharing electrons to fill up the shells, but to maintain a neutral charge. The only way to do that is by forming covalent bonds, usually with other carbons, but it could be with any other element that will form covalent bond, okay? So when a carbon share electrons with like one other carbon is going to make one bond, but it still has 3 other electrons that it can work with. Okay.
Okay.
So we see here that for it to get 4 extra electrons, it will have to make 4 bonds. When it makes,
So
hmm?
for 4 extra electrons, you said it has to make 4 bonds?
Yeah. So when it makes 4 bonds, then it's going to behave like a very stable noble gas, because all of the valence electrons are going to be filled.
Okay, let me see if I have any other questions.
Mhm.
For the mice earlier, the ones that you mentioned like were nude for testing, those were used for immune testing because they're more immune or less immune?
They don't have immune system. They are not going to attack whatever you put in the mouse. So if you want to study a tumor, for example, you can inject a tumor in the mouse, and the mouse immune system is not going to get rid of it. It's not going to have immune reactions against whatever you are injecting in the mouse because it can't. It doesn't have immune system for that.
Okay, thank you. And I just wanted to ask about the eye clickers. When we'll be able to like actually do all the eye clickers,
Yeah, I'm going to
do
give
you know?
plenty of iClicker points. Yeah, I had
Mhm.
already some points in this lecture, but yeah, I'm going to give plenty of points. So yeah, as soon as like last time we did it without a problem, and then this time all this fiasco with equipment again. But yeah, we're going to have plenty of points. And so
Okay.
okay.
All right, and just to make sure the first exam is September 10th. Well, in the syllabus, that's what it said.
Yeah, go by the syllabus. I don't remember exactly now, but yeah, I think it is.
Okay.
Yeah, it was on Wednesday, right?
Mm-hmm.
Yeah, it's September 10th. So
Okay. And you said you'll be back by next week, Friday?
yeah.
Okay.
Yeah,
All
actually,
right. Thank you so much.
I'll get out of the airport. I'm gonna go straight to class, so hopefully I'll be still alive.
Mhm.
All right.
Okay. All right. Thank you.
You're welcome.
Bye.
See you then.